Good morning. I am Dr. Joseph Kanter. I am the CEO for the Association of State and Territorial Health Officials, or ASTHO, and we are very pleased to have you join us for this scientific symposium on highly pathogenic avian influenza. I'm delighted to be hosting and moderating this event, which is put on in coordination and partnership with the Infectious Disease Society of America, IDSA, and the Council of State and Territorial Epidemiologists, CSTE. We are delighted to have with us experts from various U.S. government agencies, including the USDA, CDC, National Institutes of Allergy and Infectious Disease, as well as the FDA. Our goal for the next 2.5 hours is to delve deeper into the current body of knowledge surrounding HPAI, to discuss what is currently known, what is the data that underpins that current base of knowledge, and what areas still need to be discovered, and what work is ongoing towards that end. We all recognize that this is a dynamic pathogen, and the situation is changing quite rapidly, as has been evidenced in the news over the past week. We're happy to be able to delve deeper into the data and the scientific basis during today's session. This will be organized into 5 distinct sessions, and I will run through the topics of those 5 sessions now. We will start with a discussion on the epidemiology of HPAI among cattle. We'll go to session 2, the epidemiology of HPAI amongst humans exposed to cattle. Session 3 will include viruses, virus characterization and assessing public health countermeasures. Session 4 will focus on the development of HPAI countermeasures. And session 5 will examine milk safety. This symposium will run us about 2.5 hours, and we will take a 10-minute break in between sessions 3 and 4. I'm going to remind folks that this session is being recorded, and it is considered to be on the record. We have 12 discussants that will help us guide questions to experts from U.S. government agencies. Those 12 discussants are representing ASTHO, CSTE, and IDSA. I'm going to run through the names of those individuals now in the interest of time, so we don't waste more time going through introductions, because I know we have a lot of topic to cover. We're very pleased to have with us, representing the Association of State and Territorial Health Officials, Dr. Susan Cassandra, the State Health Officer and Assistant Secretary for Public Health in the North Carolina Department of Health and Human Services, Dr. Natasha Bagdasarian, Chief Medical Executive at the Michigan Department of Health and Human Services, Dr. Marcus Plesha, Chief Medical Officer at ASTHO, Dr. Scott Harris, ASTHO President-elect and State Health Officer at the Alabama Department of Public Health. Representing the Council of State and Territorial Epidemiologists, we are pleased to have with us Dr. Catherine Brown, State Epidemiologist and State Public Health Veterinarian at the Massachusetts Department of Public Health, Dr. Christine Hahn, the Medical Director of the Idaho Division of Public Health, Dr. Ruth Linfield, State Epidemiologist at the Minnesota Department of Public Health, and Dr. Zach Moore, State Epidemiologist at the North Carolina Department of Health and Human Services. Representing the Infectious Diseases Society of America, IDSA, we are pleased to have with us Dr. Carlos Del Rio, immediate past president of IDSA and professor of medicine at Emory School of Medicine, Dr. Shira Daron, attending physician and hospital epidemiologist at Tufts Medical Center, Dr. Andy Pavia, Chief of the Division of Pediatric Infectious Diseases at the University of Utah, and Dr. Carmen Reich, Professor at Mount San Antonio College. Again, we are delighted to host folks. We actually maxed out our capacity on this. Over 3,000 people registered, so clearly there is a hunger for information on this, and we are more than happy to convene and try and bring this information to you, namely practitioners on the ground working through this dynamic situation. With that, we'll go to our first session. Let me introduce and welcome Dr. Rosemary Sifford, Deputy Administrator and Chief Veterinary Officer at the USDA. We will discuss epidemiology of HPAI among cattle. Dr. Sifford?
Good morning, and thank you very much. It's very nice to be with you this morning to share some of the information about the work that we have had ongoing with highly pathogenic avian influenza and livestock in the United States. So I'll start this morning on the next slide with an overview of the overall situation from the animal health perspective. There has been a global outbreak of highly pathogenic avian influenza that has been ongoing for a few years now among the poultry operations around the world. The United States has been about a year behind Europe in our experience with this virus. So in the United States, our first detection in wild birds was in January of 2022, and that was followed very shortly in February 2022 with our first detection in a U.S. commercial poultry flock. Since that time, we've worked closely with states and producers to eradicate the virus from our commercial poultry flocks. We have continued this work and continue it today. We continue to see direct introductions from wild birds as those wild birds migrate across the United States. We've had over 1,100 domestic poultry facilities that we've worked with to eradicate the disease. During that time, we have had some occasional detections in mammals. Most of these mammals have been carnivorous animals that would have been expected to be exposed through their contact or consuming either dead wild birds or dead domestic poultry associated with an outbreak. But in March 2024, we found our first detection in cattle. So this was certainly a different detection in mammals from what we had experienced up to that point. And so the rest of my presentation this morning will really focus on the work that we've been doing since we had that first detection in cattle. So we have been monitoring the virus throughout our time working with poultry, doing phylogenetic analysis that's focused on the real-time analysis to assist in understanding the epidemiology, as well as to assess the potential transmission and sources of the virus. Our APHIS National Veterinary Services Laboratory uses the entire sequence genome for analysis, not just the HA gene, and has developed and refined data pipelines to aid in quality metrics and rapid identification of specific markers, such as those reported to be associated with mammalian adaptation. The IRMA pipeline provides initial assembly and assessment of quality, and the phylogenetic comparison utilizing VSNP pipeline provides a tree and table of shared unique mutations and allows us to assess the presence of markers for mammalian adaptation. Sequences for the 2022 outbreak, that is the ongoing outbreak in poultry, have been released on GSED. We recently expanded the availability of sequences by also posting on NCBI. The data sets are highly representative of the outbreak, and which over 70% of the outbreak sequences fall within 7 genotypes. Our National Veterinary Services Laboratories has worked with several partners to better characterize and understand the dynamics of the virus. HPAI is regulated by the Federal Select Agent Program, and so we comply with all of those regulations in moving materials to partners for continued work, which means that we work with entities that are registered within the Select Agent Program. So as we move on to summarize some of the epi information that we've learned as we've been working with the cattle, we were aware of an illness affecting dairy cattle across multiple states earlier this year. We became involved through our unusual morbidity and mortality event process, which allows us to help identify foreign or emerging animal diseases. The state of Texas was the state that reached out to us for assistance, and we began working in the state of Texas. We worked to sequence the virus rapidly, as well as to isolate and characterize the virus for onward sharing, as I just mentioned. We worked with our National Animal Health Laboratory Network partners to help gather more information and have personnel on site at affected dairy premises gathering additional information to assist us in understanding the virus in dairy cattle and its potential transmission. There are a few things that we have been able to identify so far. After testing in the National Animal Health Laboratory Network labs, it was identified that we do have influenza A and H5N1, Eurasian lineage goose Guangdong, clade 2.3.4.4b. This is the same clade that's been affecting wild birds and commercial poultry flocks and has caused sporadic infections in several species of wild mammals. The sequencing also identified a sporadically detected 4-gene reassortment, B3.1P13 per genoflu, that's descended from the previously predominant genotype B3.2. The B3.13 genotype was first observed in wild birds in November 2023. The few progenitor wild bird viruses were identified in the Pacific and Central flyways. The available phylogenetic analysis and epidemiology suggest a single introduction followed by secondary spread. The pathobiology of the virus in cattle and mechanism of spread are still under investigation. The whole genome sequence for virus found on recent commercial poultry premises in different states indicates it is the same strain that is affecting dairy cattle. The phylogenetic data and available epidemiological data support spread from dairy premises to poultry and then lateral spread between poultry premises. So as we further continue our work, I'll talk a little bit about what we're seeing in terms of the genotypes. This slide has all the virus sequences lined out so that you can see that all the virus sequences in this genotype are highly similar. There are 2 distinct ancestral branches with 3 wild bird and 1 skunk. That's in the red. The recent human detections in Texas is purple, and then very closely related and ancestral to current dairy sequences, but it suggests an unsampled population. All dairy samples fall in a distinct branch together with minimal genetic variation, and those are all the green branches that form the flower. There are several remarkable features of the virus that have been characterized to date. It remains very much an avian virus with no significant changes compared to other genotypes. In other words, it's not becoming a bovine virus. The viruses are highly similar, representing more of a clonal expansion versus different independent introductions, as has been seen among the majority of poultry detections prior to this event. On a distinct ancestral branch of the genotype relative to the dairy detections, and they share a common ancestor. All samples on the dairy branch, 6 SNPs not present in the skunk samples. The skunk shares 4 SNPs with a wild bird. It has 12 additional SNPs. The skunk sequence has T271A mutation associated with mammalian adaptation. Mammalian adaptation mutations, including T271A, have not been detected in dairy cattle to date. This New Mexico skunk was detected on February the 23rd and did not have the potential to be the source of the virus in the detected dairy cattle. So moving on to speak a little bit more about marker monitoring. We do continue to monitor for markers of mammalian adaptation in all of the samples that we sequence and for antiviral resistance. So far, we have not observed any from the available sequences from peri-domestic birds or cats that have been found on reportedly affected premises. And of the cattle sequences that we have sequenced, and at the time this slide was produced, there were 260. There are more now, but with the sequences that we have reviewed, we've only found 1 that has had a mixed base population at 627 in the PV2 gene. And that is the only indication that we have had in any of our sequencing work of any mammalian adaptation markers. So I'll compare this a little bit to what we've seen on the poultry side. This slide shows a little bit from our poultry detections. And you can see that we have here some of our recent poultry detections that are on a distinct branch. So in the orange is a Texas herd Texas flock that was identified on March the 16th of 2024. And then in the more pink are other Texas herds that were identified in March and early April. These are interspersed with the dairy detections that are in green and blue here, Texas 07 in green, and Michigan 001 and Ohio 001 in blue. So this indicates that there may be a potential for common source or lateral transmission in either direction between these flocks and herds. And given the timing of identification and clinical signs, we believe and understand that it was most likely from the dairy herds back into the poultry flocks. We continue to look at the exact movement that might have been supportive of this kind of transfer of the virus from one to the other. So we do continue to look very closely at this virus and the way it's working in livestock and try to understand it much more clearly. We continue to conduct testing and analyze the sequencing results. We have epidemiologists, and we're working closely with our state counterparts and their epidemiologists to administer questionnaires to producers to better understand movements and activities that might have led to virus entering a facility and then or maybe moving around or off the facility. And we really appreciate all of those producers who have participated in answering all these questions. Just yesterday, we issued a federal order intended to minimize the risk of disease spread and help us to gather some additional information to better understand the situation and make scientifically informed decisions. That federal order requires reporting of positive influenza tests in livestock, requires mandatory testing for influenza for dairy cattle before interstate movement, and then places movement restrictions on any dairy cattle that are positive for avian influenza virus. We also included that positive herds must undergo an epidemiological investigation and movement tracing, again, to help us gather as much information as possible about this current situation. We continue to collaborate with our partners to validate different sample types, to do serological and to develop serological and pen-side tests, and to consider additional testing options. We're conducting transmission studies in cattle in partnership with our colleagues from ARS at the National Centers for Animal Health and collecting additional environmental samples for further insight into the disease and its spread. FDA and FSIS are conducting milk and meat safety studies and reiterate the safety of pasteurized milk and meat products. USDA is sharing the results of our ongoing investigation with many partners. This includes our state and federal animal health and public health organizations. We have made our gene sequences and virus isolate available through the Center for Veterinary Biologics to support additional research. And as I've mentioned before, the sequences are uploaded into GICID and NCBI. Trading partners have requested additional information on our epidemiology, and we are responding to their requests for information as we receive them to provide information that assists them in mitigating any trade impacts. And then, of course, we've been reporting to WOA through our standard reporting process and sharing information with the WOA community to help other countries prepare for potential animal health situations for them as well. With that, I will open it up for questions.
Dr. Sifford, thank you so very much. We know you and your team have been working extremely hard the past few weeks. Please know that we deeply appreciate you. We now have about 10 minutes of question and answer on this session. I'm going to first turn to Dr. Susan Cassandra from North Carolina. Dr. Cassandra.
Thank you. Thanks, Dr. Kanter, and thanks, Dr. Sifford, for the information and for all USDA is doing. I had a question going back to what you shared, starting with, we understand that this was likely one event from poultry to cattle that led to the introduction into cattle. Can you talk a little bit more about what we know about the spread between cattle? Do we have a sense of how that transmission is occurring and what some of the studies are to better understand how that viral transmission is occurring between cattle?
Yes, we do have some information. It is still early, and we do still continue to gather information to make sure that we can understand as completely as possible how the virus is moving. But what we can say at this point is that there is a high viral load in the milk in the cattle, and it appears that the transmission is happening mostly within the lactating herd. So that leads us to believe that that the movement through the cattle is most likely associated with milk, whether that be through the actual process of milking or just from contamination from milk from a cow with a high viral load to other cattle. In herds that are outside the geographic region that appears to be where the first spillover event happened from birds into cattle, those herds are very consistently showing epidemiological links to herds within that geographic region. So we believe it has moved with the cattle, with lactating cattle from those herds into the other herds. For this reason, we are strongly encouraging producers to limit movement of cattle, particularly lactating cattle, as much as possible. And as I mentioned yesterday, we just implemented the federal order to require testing prior to interstate movement.
Thank you, Dr. Sifford. Turn to Dr. Catherine Brown, Massachusetts.
Thank you so much, Dr. Kanter and Dr. Sifford, just echoing the appreciation for all of the work that you're doing. There are a lot of questions that need to be answered very quickly, as you are clearly working on. So building a little bit on the previous question, I mean, there's a lot to learn about the transmission between cattle and then cattle back to birds. I'm just wondering about your thoughts about surveillance in other mammalian species on farms and potentially both domestic and wildlife as we think about sort of this new opportunity for transmission. And just quick second question, how much virus is in being shed from the GI tract of cattle, since that could speak to environmental contamination?
Yeah, on the surveillance in other mammals, we, as you know, have a very strong surveillance system in poultry. We also have a surveillance in our feral swine. Our Wildlife Services organization started several months ago to monitor the feral swine population. So far, no indications of any virus in that population. And while we don't have a specific surveillance program for avian influenza in swine, we do have an influenza program for domestic swine. And our expectation is that any avian influenza would be picked up through that influenza program in swine. We do continue to share messages with the entire producer community to have other producers be monitoring for any evidence of disease and to please report any sick animals for further diagnostic workup. This message has been particularly important, I think, for beef cattle producers. Beef cattle are managed in certainly a different way that may change their risk profile, but we certainly want them to be monitoring for any evidence of clinical signs.
And just quickly, is there how much virus is being shed from the GI tract of cattle?
Thank you for repeating that question. From the samples that we have so far, which admittedly is a small number of samples from the GI tract, we do not see any significant shedding. We've sampled different tissues from cattle provided to us from FSIS, and we're seeing still concentration in the milk and the mammary tissue as the highest tissues where we see viral concentrations.
Great. Thank you, Dr. Sifford. Dr. Del Rio, Emory University.
Thank you so much. That was really an excellent presentation. I think one of the, I think clarifies a lot of things. I think one of the things that is asking the chat that I was wondering myself, is that the human isolate appears somewhat different than the cattle isolates, and somewhere between sort of skunk wildlife and the cattle. And how does this information inform the hypothesis as to how does a human infection occur? What is the current hypothesis? How does human infection occur? And what is USDA doing in collaboration with local partners as far as testing of people working in this industry and knowing whether we're seeing asymptomatic cases, we're seeing other infections? Because there seem to be reports of people working in the industry having symptoms, but we know very little about surveillance and how this, sort of human, animal to human, bridge is being monitored.
Yeah. Yes, the fact that the human who was sampled has a sequence that appears to be a predecessor to those that we have found in cattle. Our interpretation at this point is that means likely that person became infected through a herd that we did not receive any samples from for testing. As I mentioned, there were herds that had clinical signs in that geographic area before we had the first herd that we had positive H5N1 test results in. And so it is that we think is the strongest hypothesis at this time with regard to the infection for that person. We collaborate closely with CDC and local public health for the human testing. So we are not directly involved in that. We share information with them to allow them to work with those farmers and farm workers to gather up additional information and conduct any necessary testing.
Thank you very much.
Thank you, Dr. Sifford. I'm going to go back to Dr. Cassandra.
Great, thanks. Dr. Sifford, can you talk a little bit about what the risk is to beef cattle? You mentioned that the virus is being found in high levels in milk and mammary tissue. And so can you talk a little bit about the risk to beef cattle, not dairy cattle?
Yes. As I mentioned, we believe that originally there was a spillover event from wild birds into the dairy cattle. Likely, this has to do with the management of dairy cattle being in much more confined spaces, and that confinement and the access to feed and water attracting wild birds to the facility. So if we were to see that kind of spill over into beef cattle, we would expect that it might be more in a feedlot environment or some environment where, again, the cattle might be more confined with open access to feed and water that might attract the birds, less so in a cow-calf operation where the cattle are more spread out and there isn't as much to attract wild birds. We have been working closely with those producers to identify any clinical signs that they see and have not had any clinical signs reported to date. But we believe that the management of those cattle, as I mentioned, being significantly different than the management of dairy cattle, probably changes their risk profile significantly as well.
Great. Thank you, Dr. Sifford. We have a couple more minutes for further questions. Dr. Brown.
Yeah, thanks. I know that transmission studies with the cattle are ongoing, but do you have any early information about how long cattle shed virus?
At this point, we do not have very much information with that regard. What I can say is that we have had one herd of cattle that did not have any clinical signs that tested positive. In that herd, cattle had been moved into that herd from a herd that had that subsequently had clinical signs in positive cattle. However, the herd that received cattle to date has not had any clinical signs. So we do know that the virus can be present in cattle not exhibiting clinical signs, but we are just getting underway with those studies to give us an idea of the opportunity for viremia either ahead of or after clinical signs. So we should have more information about that in the coming weeks.
Great question. Thank you. Got time for maybe one more question on this session. Do any of the other discussants? Yep, Dr. Moore, I see your hand up. Go ahead.
Yeah, thank you so much for the great presentation. I wonder if you are hearing or have any concerns about real or perceived disincentives for participation in testing beyond what's now required for interstate travel, and if there's anything that USDA is working on to address concerns about economic or publicity issues that may arise.
Yeah, I mean, we certainly only have anecdotal information regarding any concerns that might be expressed. We have tried to address those by assuring folks what the process is for testing and what they can expect from that process. Producers are seeing that cattle will be sick off feed and with significantly decreased milk production for a week, a little longer than a week, and then appear to either fully recover or recover very nearly to their full production before they were sick. And so, you know, we have heard anecdotally that for some producers, you know, that's a short enough window that it doesn't encourage them to share samples with us, but we have been sharing our message broadly that we need those samples, and we have continued to get samples from herds across the states. So at this point, we are trying to address, again, what we hear is anecdotal reluctance by just sharing more information about what producers can expect and what our test results might mean for understanding how to avoid this potential event in the future and put proper mitigations in place.
Thank you. We have time for one final question on the session. Dr. Bagdasarian, Michigan.
Thank you so much. Just to piggyback on the last question from Dr. Moore regarding interstate movement of cattle, there still does appear to be quite a bit of movement within states of cattle. Is there any consideration to testing or additional requirements before moving cattle within states from facility to facility?
Yeah, at this point, we're working very closely with our state animal health partners for their authorities in that space. And so if we do identify an affected herd in a state, working very closely with the state animal health official for what procedures we can put in place to restrict movement from those herds.
Let's give up. Dr. Sifford, you froze up. Do you want to finish that thought if you're with us? Yeah.
Sorry about that. Yeah, just I was saying that we are working closely with the state animal health officials to ensure that that we use the authorities they have available to them once we have an affected herd to restrict movement from those herds.
Great. That will conclude the first session. Dr. Sifford, thank you so much, and please extend our gratitude to your team. We're deeply appreciative of the work they're doing on this. It really does make a difference. So thank you, and thank your team as well. We will now transition to the second session, examining the epidemiology of HPAI among humans exposed to cattle. We're very pleased and grateful to have Dr. Sandra Olson. She is Associate Director for Preparedness and Response of the Influenza Division at the CDC. Dr. Olson?
Thank you. Thanks very much, and I appreciate the opportunity to speak to everyone today. I'm going to shift gears now and talk a little bit about the epidemiology. And just to start, I wanted to give a brief background on novel influenza viruses. These are subtypes of influenza A viruses that are different from currently circulating influenza viruses in humans. And so they can include a number of different subtypes, including what we're discussing today, H5 viruses. They can also be H1 and H3 subtypes if the virus originates from a non-human species or is the result of a genetic reassortment between animals and humans. Each novel case of influenza is reportable to WHO through the international health regulations. And from a public health standpoint, it's critical to investigate each case to really understand how a person became infected, and to make sure that there aren't any other cases, and also to prevent onward spread. In addition, these viruses can cause severe disease, so prompt treatment is also important. Sporadic cases, however, do not always turn into more, and certainly not all turn into a pandemic. So to put things in a bit of a historical perspective, H5N1 high path virus was identified in domestic waterfowl in China in 1996, and the first human case was reported in 1997. So this slide shows all of the human cases reported since 1997. There have been 909 reported from 23 countries. And about half of these, 52%, of the human cases have resulted in death. And that's just a reminder of why this disease is such a public health concern. These cases of H5N1 reflect sporadic cases, and you'll see a large spike in this graph in 2015. It's the orange bars. Those were cases reported from Egypt, and it's around 350 cases. And these were likely due to these additional infections were likely result of poultry exposures and more human exposure to these poultries. Around 70% of those cases were exposed to backyard birds. But since 2015, there are only a small number of human cases reported. Between 2004 and 2007, we did see rare, limited, non-sustained instances of human-to-human transmission that's most likely occurring in small number of family members after prolonged close exposure that was unprotected to a symptomatic case. The highly pathogenic avian influenza H5N1 viruses occur mainly in birds, and they're especially fatal in poultry. In 2020, the clade 2344B first emerged, and that's the virus that's currently causing wild bird and poultry outbreaks in the United States and other countries. So this slide shows a USDA map on the right and reports the detections in wild birds by state. And you can see that it's widespread over the United States. The graphic on the lower left shows the summary of the human and animal detections, including wild bird, poultry, and now dairy cattle. And I think this slide highlights that poultry continue to be the most heavily affected animals, with over 90 million affected in this current episodic. Since 2022, there have been 26 human cases of H5N1 virus infection reported to WHO, and half of those were known to be associated with the clade 2344B. The dairy cow illness that began in early 2024, you just heard a lot about, so I'll make this part brief. The cows have shown significant decrease in milk production and quality, and impacted farms in Kansas and Texas were the first reported states, with herds being infected there. And as of today, there are affected herds in 33 farms across 8 states. And there are also 6 detections in cats confirmed on various dairy farms, and also wild bird confirmations on some of the farms. One human case was recently reported. This was on April 1st. The state of Texas announced that a person had tested positive for H5N1 virus. The preliminary testing was done at a public health lab in Texas, and it indicated that the specimen was presumptive positive for influenza H5 virus. CDC then confirmed the specimen as H5N1 using diagnostics, real-time PCR diagnostics and sequencing in less than 24 hours after receiving it in Atlanta. The person who who was positive in Texas had direct exposure to cattle, as you heard, that were presumed to be infected with high-path avian influenza. Fortunately, this person reported eye redness as their only symptom, consistent with conjunctivitis, and was provided treatment and has recovered. There were also no reports of illness in household contacts. This continues to be the only case reported from someone exposed to infected cattle. And a reminder, this is the second detection of avian influenza virus in a person in the United States. In 2022, a case was associated with infected poultry exposure, and the person also had very mild illness and recovered. As with other H5N1 cases in the last few years, there's no indication of any human-human transmission. Given the detection of one human case of H5N1, there's a need to know what's going on more generally. So I will walk through how CDC monitors H5N1 infection in people in the United States. CDC's existing influenza surveillance systems and laboratory infrastructure are well equipped to rapidly detect human cases of H5N1 virus in the US. We collect, compile, and analyze information on influenza activity year-round, and this is done in collaboration with many partners at the federal, state, and local level. As part of our response, we're actively reaching out to affected states and holding technical assistance calls. These calls are helpful both to share information and share guidance, and for us to learn as well more information about the outbreak. We're also working with states to implement monitoring of persons exposed to infected humans, including poultry and cattle. And then we're conducting enhanced surveillance through a number of systems and also planning epidemiologic studies. We started monitoring for persons exposed to infected poultry back in 2014 when several different H5 viruses, H5N2, H5N1, and H5N8, were detected in poultry and wild birds in the United States and Canada. And at this time, we worked closely with USDA and developed joint monitoring guidance. Our guidance recommends to monitor people exposed to infected birds and poultry or other animals for 10 days after exposure. In these events, the monitoring approach was fairly straightforward since poultry are being depopulated, so the exposure time is known and somewhat limited in scope. Local and state health departments and CDC have worked with these operations and also with USDA responders to conduct the monitoring. Since the current episodic of 2022, together with our partners, we have monitored over 8,400 people in 52 jurisdictions. About 200 of these persons reported symptoms and were tested for novel flu. As I mentioned, there was one human case in 2022 associated with infected poultry exposure. In the current cattle outbreak, so far there have been 44 people being actively monitored, and additional persons are being passively monitored. In other words, they monitor themselves and report if they develop symptoms. So far, states and CDC have tested at least 23 people, and there was the one case identified in Texas. And here I want to acknowledge that the situation in cattle is a little different than that in poultry. If dairy cattle are ill and we have known exposure, though we know that the illness in cattle can go on for several weeks. So that puts workers at an ongoing risk, and thus the period for monitoring will be longer. In situations where the cattle are not showing signs of illness but testing positive, it gets a little less clear on how to define this exposure period. So these are just examples of some of the challenges we're working through and highlights understanding why knowing the routes of transmission from cattle or the environment or milk is so important to be able to begin to make informed decisions about the risk to humans and how best to protect workers. Going back to our broader surveillance, CDC's existing influenza surveillance systems and laboratory infrastructure are well equipped to rapidly detect human cases of H5N1 virus in the United States. We collect, compile, and analyze information on influenza year-round, and again, this is done with many partners. Human cases of novel influenza are nationally notifiable, so every case is investigated and reported to CDC. And data of all reported cases, novel cases, are also available on our website. In addition, public health laboratories use CDC's diagnostic tools to detect both seasonal influenza viruses and novel viruses. These tools are used at more than 100 public health laboratories in all 50 states. So the graph on the upper right corner of this slide shows you those data. And we get information on the virus type and subtype, and a sample of these viruses are sent to CDC for further characterization. This system is right-sized for novel event detection. So we have a goal of detecting a novel influenza virus among positive samples across all jurisdiction. And we set a low enough threshold so that we can implement effective prevention and control measures. We also monitor changes in the test positivity for influenza in the clinical setting, and this is done through about 300 clinical labs in the US. And this is the second graph on the lower right. The black line shows the overall percent positive of specimens, the percentage of specimens tested that are positive for influenza. So by both indicators on those two graphs on the right, public health labs and clinical labs, flu activity in the US is currently going down. And that's consistent with what we know about flu seasonality in the United States this time of year. We're also looking at the clinical syndrome of influenza and are using data from the National Syndromic Surveillance Program, which collects, analyzes, and shares electronic data received through multiple healthcare settings, including emergency departments. These data also show a low percentage of ED visits for flu-like symptoms, and that's the graph at the bottom in black and white. So together, these systems give us good insight into flu activity in the US. And more importantly, right now, they're reassuring that nothing unexpected is happening. In addition to these enhanced systems for flu, we recently explored the question of whether our general health system in the US would pick up novel cases occurring in the community. This was done by simulating a model with different inputs exploring 3 healthcare settings: outpatient urgent care and emergency room departments, inpatients, and intensive care units. And this work asked the 3 questions. What's the probability cases would be detected? Which healthcare settings may be more likely to pick up cases? And how might things change with increased disease severity, which could vary for H5? And before I go on to show you the results, I'll just say that this is a paper that is available online at the link below. Next slide. What we found, to no surprise, is that detection of cases is dependent on clinical severity. So as severity increases, the probability of detection also increases in all health settings. And this is due to the fact that the more severe you are, the more likely you are to require medical attention. So for example, assuming severity is equal to recently observed worldwide H5 cases, the median probability of detecting at least one case is around 90% in the urgent care setting and 100% in hospitals and ICU. So how is this relevant to the current response? Well, first, it demonstrates that our systems are robust. Second, it also reaffirms that that a critical part of this is that clinicians maintain high rates of testing in persons with pertinent exposure history and forward those specimens on to public health laboratories for further virus characterization. The last area I want to touch on is epidemiologic investigations. CDC is currently prepared to deploy teams to several states to aid in investigations, should there be interest. We are working with health and agricultural partners at the local, state, and federal level, as well as with affected farms. We feel these types of investigations are critical and can help us answer important public health questions. And those questions would include, is there evidence of infection of H5N1 in exposed populations? So we know about the one case. Are there others? If we find infections, what is the spectrum of illness and the rate of asymptomatic infection? What are the types of exposures? So we talked a little bit about this before, but is it direct exposure to cattle? Is it exposure to raw milk? Is it exposure to the environment or something else? Knowing that information is critical. Are there behaviors associated with human infections or things that we can learn to protect someone from infection? Also, we want to assess the risk of symptomatic and asymptomatic infections, and this would be done through both specimen collection as well as survey data to ascertain these exposures. Next slide. We've conducted studies like this in the past. For example, this is a recent example from 2022. During the poultry response, we worked with states to assess the risk of symptomatic and asymptomatic infection. And we did this study through a survey to ask questions about exposures and by collecting specimens. In this example, over 4,000 people were tested and monitored. And these were people that were exposed to birds. 134 or 3% experienced onset of one or more symptoms compatible with influenza infection. These were all mild. And those had specimens collected, respiratory specimens collected for diagnostic testing. And there was one PCR positive. That's the case I've mentioned before from 2022. And in a subset of this, there was also a serologic component to look for antibodies in the sera of people, and there were no serologic positives found. So these two types of studies are critical to understand the basic epidemiology of a new virus. How is it being transmitted? What is the risk to humans? What can we learn to best inform our guidance? CDC is proactive at communicating information about the response and providing technical guidance to our partners. And as I said, it's critical to have the best available data to inform this guidance. These documents now include more specific information that are tailored to farm workers and to the specific outbreak. So for example, recently we published a health alert network health advisory to inform clinicians, state health departments, and the public about updated information on the human case and to emphasize our key recommendations. We've also published a series of updated guidance documents on our website. We've updated recommendations for people who come into contact with infected animals, including farm workers. We also have guidance for clinicians on monitoring, testing, and antiviral treatment of patients with avian influenza virus and of use of antivirals in exposed persons. And finally, we have recommendations for surveillance and testing, as well as infection prevention control. And as I said, the antiviral treatment. So to summarize, we have one human case associated with the current outbreak of H5N1 in dairy cattle. We are actively working with our partners to monitor exposed persons, and that remains a priority. We would also like to conduct some more in-depth epidemiologic studies to help us understand the risk of persons and better identify ways to prevent additional cases. We feel that ongoing collaboration between all the disciplines, animal health, environment, is really critical, and at all levels, the federal, the state, the local. And just to reiterate that this is not a new process for us. For us, this is a new way cattle the virus can spread and infect people. We rely on the decades of processes and data to help us investigate the situation and understand the potential for spread. So I think I'll stop there and see if there are any questions.
Thank you so much, Dr. Olson. That was an excellent presentation. We have about 6 minutes left for questions. Let me turn first to Dr. Hahn, Idaho.
Yeah, thank you. Thank you so much for that. There are so many questions, but my first question for you, or my question for you is, in light of the fact that right now so much of this activity is in the animal world, of course, investigating the cattle, transmission between cattle and other animals, can you help us understand what is CDC looking at as far as indicators for human risk to humans? Right now, we believe human risk is low. We keep hearing that. All the evidence right now is saying that the risk is low. There's only been one human identified, but only 23 people have been tested. What information does CDC need, and we in public health need, to help gather information on risk to humans. So whether that's evidence from milk that I know FDA is working on right now, evidence in humans, and how, given the challenge of testing and getting enough tests done, how can we be assured that we're getting that information?
Yeah, thank you, Christine. I think that's a really good question. And I think to answer your question, I think all of that information is important. And I think for us, trying to do these types of a little bit more in-depth investigations to get at specific exposures is really critical. I think right now, we know the virus is in high loads in milk, but we don't know if it's the exposure to milk that's, the risk to humans. We don't know if it's fomite transmission. We don't know if there's aerosolization of the milk. I think we have a lot of questions. And so I think to get at that, we, are very interested in trying to do these more in-depth studies to get at those types of questions. But I think the whole picture is what's important. So understanding, how it's being spread between animals, what is the contact that people have with cattle or other animals or the environment on the farm, sort of delineating all of that is important. And understanding if maybe there's other mild illnesses that aren't presenting to care, that all of that is what we would hope to ascertain by doing a more in-depth study. Over.
Thank you. Dr. Bagdasarian, Michigan.
Thank you so much. That presentation was excellent. And I'm so glad to hear about the additional studies that will be taking place. I think that you're absolutely correct that there's so much we have to learn about the interface between humans and dairy cows, after most of what we have learned has been this interaction between humans and poultry. I'm wondering, as we talk about humans exposed or humans who could be at risk, I think we've mostly been talking about folks who work at dairy farms. But we're finding out that there are also lots of people involved in processing the milk, taking products on and off the farms. Is there a plan to broaden that scope and look at other folks who may be coming into contact with sick animals or infected material? Thank you.
Yeah, I think that's another great question. Thank you. We recently updated our guidance on protection for workers to broaden it to answer just this question. I think, is there a risk of other workers as well, such as people working in a slaughterhouse? So I think, we don't know, but I think we would suspect that if there's widespread virus or contamination that, on a farm, that there may be other workers that come into contact. And so I think, it's important to get the guidance out about how to protect yourself, because we do feel that you can protect yourself from this virus. And that's important as we learn more about the ways that the virus is transmitted. So I think both of those sort of go hand in hand, learning more and protecting workers, which is now a broader group, I think, as you point out. Over.
Thank you, Dr. Olson. Dr. Derone, Tufts University.
Thank you for that wonderful talk. My question is about testing. I direct an infection control program at a hospital system, and I can envision a scenario where even if there's no change in the epidemiology or risk of person-to-person transmission, if we have a bad flu season, say this fall, the number of people with influenza A and animal exposure, or who otherwise meet whatever our person under investigation criteria are, could be large. And our experience from early in COVID tells us that sending specimens to a regional public health lab or CDC is associated with long turnaround times, which gets longer as that volume of testing increases. Most patients do not have a private room at home in which to isolate. I'm worried about a backlog of patients who we need to keep in the hospital on isolation, waiting for test results again, based on my experience with early COVID. I don't think we're there, but what would have to be true to get to a point where we would have commercially available PCR testing and or home testing for H5N1?
Yeah, I mean, we've definitely thought about this, and I think, as you said, we're not there, but it's always good to think about. And I think there are, several several indicators that would, that we look for that could make us concerned if the virus started changing, if we saw, more clusters of disease, or if we began to see human to human spread. There are a number of indicators we would look for. And I think that, we have thought through this testing question, and there is a dialogue with commercial manufacturers, a number of folks that are interested in doing this. And I think that if we feel that we, meet a threshold, that we would begin to make those shifts. So I think, yes, we have learned a lot from recent experiences, and I think are aware of that. And I think from the epidemiologic standpoint, we're looking for, that's why this epidemiologic investigations are so critical, is to really understand what's going on and feel confident that nothing is changing, or that we're not missing anything. Over.
Thank you so much, Dr. Olson. We deeply appreciate your efforts, and your team as well. This will conclude the second session of our symposium. We will now move on to session number 3. This session will examine virus characterization, and will assess the public health countermeasures that are currently in play. I'm happy to introduce Dr. Vivian Dugan. She's the director of the Influenza Division at the CDC. Dr. Dugan?
Thank you very much, Dr. Kanter. I really appreciate this opportunity to talk to you a little bit more about what we know about this H5N1 virus from the individual in Texas, and how CDC has used that data from a genetic perspective, and also some of the phenotypic assessments that we've performed on this particular virus, and kind of where we are positioned in a kind of our pandemic preparedness and readiness posture. So picking up a little bit from where Dr. Olson just left off, I'm going to focus about what we know from the sequence data generated very rapidly at CDC from this human case in Texas of H5N1. So one of the first things, of course, we look for are what is the current impact to our existing diagnostics, and also our current treatments, as well as candidate vaccine viruses that we, along with several other groups at the World Health Organization, collaborating centers, generate on a regular basis. And so the good news is that, of course, this is the data are publicly available. There's no impact to our current diagnostics, our treatments, or the candidate vaccine viruses. The H5HA, the hemagglutinin of that human virus, was very closely related to 2 available candidate vaccine viruses that were generated here by CDC, WHO Collaborating Center laboratory leaders. And so from just the sequence data, we can expect and anticipate that those CVVs would provide good protection against the virus. Good to keep in mind that these candidate vaccine viruses are the precursor to moving into large-scale vaccine production, should that be needed. But when we look at all of these data together, and a little bit more data that I'll show on the next slide, with the current epidemiology, again, this has been mentioned, that our assessment by CDC right now of the risk to the general public remains low at this time, but we're always assessing this as new data become available. So when we dive down a little bit deeper and look at the sequence data, we did obtain sequence data from the conjunctival specimen, but not the nasopharyngeal specimen from this individual. And that, to our experts, suggests a lack of a respiratory infection. And it also aligned with the cycle threshold, or CT values, that were obtained by real-time RT-PCR on the specimens for 18 CT value for the conjunctival specimen, and 33 CT value for that nasopharyngeal specimen. And when we analyze the H5 hemagglutinin gene segment, it is that clade 2344B. You've heard a little bit about that, where it's circulating, and the impact on this particular H5 clade in poultry and other animals. And so when we analyze and compare the human virus sequence, we've noted that it's nearly identical to those that were obtained and shared by our collaborators at USDA from infected dairy cattle and birds in Texas. And so to our experts, again, our phylogenetic analyses and evolutionary virologists, this looks like it's primarily an avian virus. It has avian viral genetic characteristics, and it also lacked any changes that we would expect to see in the genetic code for adaptation to better infect mammals. That said, it did contain one marker, that one substitution in the polymerase II protein or gene at position 627. And so a little bit about this. This particular substitution or mutation has not been found in Texas dairy cattle or birds. It is not currently circulating in wild birds or poultry, and it may have been acquired in the individual, in the patient, during the development of conjunctivitis. It has been previously identified in other human cases of H5N1, so it's not surprising and it's not uncommon, although it has been associated with mammalian adaptation in experimental studies. There's no evidence from this particular case that there was onward transmission of this virus among humans after the infection containing this mutation. So I'm going to go a little bit into the hemagglutinin, the HA protein. And so this gene codes for one of the two surface glycoproteins on the virus. And this is really a central and important species specificity for the HA, because it's responsible for virus attachment and fusion with the host cell. And so the HA, or hemagglutinin, is the primary target of neutralizing antibodies elicited by infection or vaccination. And so generally, the hemagglutinins of human viruses preferentially bind through this alpha-2,6 linkage. And so this is a frequent type of linkage in human respiratory epithelial. In contrast, the hemagglutinin or HA of avian strains bind differently. They bind preferentially to the alpha-2,3 linked sialic acids. And so these are more abundant in the avian intestinal tract. So that's all to say, when we look at the structure and 3D protein structure of the hemagglutinin of a nearly identical H5HA shown there, we look at that receptor binding site, or the RBS, and we compare what the Texas virus had to other known avian viruses that are identical in the same space. And so this shows the receptor binding preference analysis using kind of a proxy virus, using two different types of arrays. And you can see there the blue and the red, the avian type glycans versus the human type glycans, with those top bars basically showing the receptor propensity to it. And so when we look at the different glycans and linkages, while one looks at the preference for glycan length, the other looks at single chain or branch glycans. It gets very technical, but essentially both revealed a strict preference for the alpha-2,3 salicide. So that's the avian type glycans. And so this basically we'll be working on this more in the phenotype space, but our expectation that is this the same receptor preference will hold true for the H5 from the viruses in the current outbreak. And so these avian influenza viruses do not have the ability to easily infect and bind to the alpha-2,6 or more human preferential binding. A little bit about our testing for novel influenza A viruses. As Dr. Olson mentioned, the novel infections in people of influenza A are nationally notifiable, and we investigate all of those cases. And so another thing, again, looking at the genetic sequence of the Texas virus, we found no genetic changes that would impact CDC's real-time RT-PCR diagnostic assays that are available at U.S. public health labs, and they're also used year-round. That said, when we think about commercial tests, which is often an important question, These tests for influenza A are widely available, and we expect that they will detect an H5 virus as an influenza A. They may not be subtype specific. That said, I think it's also an important note following up from the last questions that we are actively partnering with commercial diagnostic developers and testing companies in case we need to scale up to increase that testing if that is needed. So we are positioning ourselves to be really ready to go should the situation change. Another thing that we do is to look at antiviral treatments. And so when we, in addition to using the viral sequence data to assess our countermeasures that are currently available, we look at the phenotypic attributes. So does this particular Texas virus react to any of these antiviral medications? And so we found that and confirmed really even most recently yesterday that both our neuraminidase inhibitors and our polymerase inhibitors would be effective against this Texas H5 virus. And then that work is ongoing. So as more data become available, more sequence data, we're iteratively and constantly assessing and checking the genetic sequence data to look for any changes in the genetic code of the viruses that might impact our ability to use these countermeasures. And so these data would currently are very supportive that antiviral treatments would be effective against H5N1 viruses should the situation change. And again, that continued in silico and then in vitro assessments are really critical for our antiviral preparedness. So in the past few years, and even prior to that, we've really made a concerted effort to invest on our influenza genomics and analysis capacity in the US. Currently, CDC sequence about 6,000 seasonal influenza viruses each year. But we also have, through our partnerships and also through APHL, we have funded 3 labs in California, New York, and Wisconsin at the state public health level that serve as national influenza reference centers. And these labs mirror a lot of what we do here in Atlanta in our labs. And so every state sends specimens, influenza specimens, to one of these 3 labs. They are equipped to perform genomic sequencing as well as virus isolation. And so that's one of our kind of major arms of activities and preparedness in the seasonal space that also serves in our pandemic preparedness space. And then recently, we actually invested in influenza sequencing centers at those 6 states listed there. Really to, again, kind of invest in our preparedness and sequencing capacity and in terms of pandemic readiness. And so these groups are all ready to serve and provide a minimal, sustainable level of testing that's needed to maintain preparedness for pandemic response should the situation change and in the event of a widespread novel outbreak. Quickly moving to our global surveillance, this is really central to the prevention and mitigation of both seasonal and pandemic influenza. The CDC serves as one of 7 WHO collaborating centers. We play a major role in year-round surveillance for early detection and identification of, again, these kind of distinct and drifted seasonal viruses, as well as novel influenza A viruses that may have pandemic potential. And so we collect and analyze the data from these different types of viruses from around the world, looking at their epidemiology, their genetic and antigenic changes, their antiviral susceptibility, and of course, genetic characterization. And so this is often where the development of our candidate vaccine viruses is coordinated through. It remains an essential component for the overall global strategy for influenza pandemic preparedness. And so to give you an idea of what the diversity looks like of just the clade 2344B viruses, these are just kind of a snippet of that diversity. As you can see, it's quite distinct. There's quite a number of changes that have happened from 2344A all the way to 2344G. With some geographic restrictions or diversity there. What's important to note, and that red text that's very small on the slide, you'll see in the next slide, these are the CVVs that have been made as a result of all of the World Health Collaborating Centers really putting their brains together and making sure that we have candidates to go depending on what may or may not emerge that represent antigenically different viruses. So this is a snippet of that library of the few candidate vaccine viruses that are relevant for the 2344B and other clades. So CDC and WHO collaborating centers generate these candidate vaccine viruses. At CDC, they are made under good laboratory practices. They are available to vaccine manufacturers and listed on the WHO website. So we did a little bit of preliminary antigenic characterization using ferret antisera. And so this preliminary data is basically of the Texas virus shown there at the bottom as a test antigen. And basically the bottom line is that it showed really good cross-reactivity against ferret antisera raised against the existing clade 2344B candidate vaccine viruses. And those two, as you can see, are very long names, but they are shown there on the left-hand side. And then of course the sera is there at the top. And so in the bottom right-hand corner is really what you're looking at. A geometric mean titer, you're looking for a titer greater than or equal to 40, which is considered a correlate of protection. And so you can see those two Texas viruses grown in cell or eggs had 40 or more geometric mean titers to those 2344B candidate vaccine virus sera generated in animals. So very briefly talking about assessing the potential pandemic risk. I mentioned earlier, and we've mentioned throughout, that CDC continues to support that the risk to the overall general public is low. That said, we also have a tool that we use to assess pandemic preparedness and really looking at the risk of a potential pandemic occurring. And so this tool is called the IRAT, the Influenza Risk Assessment Tool. Again, it's to assess the potential pandemic risk. And it's really a tool that we use to evaluate and prioritize our resources across the US government in thinking about many different viruses that are in animals and may move into people. And so when we run an IRAT, we generate a score. It's through those 10 criteria that are shown there on the slide. And they're evaluated by subject matter experts to really rank a particular prototype virus for all of these different areas, these different 10 risk elements. And so an emergence score and a public health impact score are where this lands. And so I just want to mention that these particular 10 criteria require data. And so we're actively working on this to assess the Texas virus. So that's ongoing. And so more to come in this particular space, but it does take some time to generate the potential pandemic risk score. And then kind of second to last slide, I just wanted to talk a little bit about the flu risk management meeting. So this is a pretty well-established US government interagency group of senior influenza experts from a number of different agencies across the US government. It was established over 15 years ago, really to share information and to make recommendations for, again, monitoring, assessing, and really looking at circulating influenza virus strains that have pandemic potential to really protect the American public. And so it's a really fantastic team of experts. They're actually meeting right now to really talk about some of these pre-pandemic vaccines and surveillance, as well as seasonal, as well as influenza therapeutics and diagnostics. So overall, a summary, the risk remains low at this time based on a lot of the things that I've already talked about. We continue to be in a strong readiness posture at CDC as new data become available. We'll be doing additional phenotypic characterization, looking at receptor binding, population serology is another activity we have planned. And as those new data become available, going from that genotype to the phenotype, it's really central to our preparedness efforts. And then also continually looking at the risk assessment, not just the influenza risk assessment tool for potential pandemics, but really understanding the current public health risk that we would immediately be messaging about should we see a change that would alert us in this space. Just want to thank everybody for the ongoing work that we're doing in the interagency space. It's been really critical, partnering with USDA, ASPR, NIAID, and everyone, FDA, and in the space to really make sure that we are sharing information in real time and getting and staying the most up to date as we can. Happy to take questions.
Yeah, Dr. Dugan, thank you so much. That was an excellent presentation. And thank you as well for highlighting the interagency cooperation. I think that's important for people to hear and understand. We have about 5 minutes for questions. Let's go first to Dr. Marcus Plesha.
Thank you very much, Dr. Dugan, for your presentation. I particularly liked your slides towards the end where you kind of walked us through the things that might make us more concerned that that this could become more of a risk to humans. I thought that was a really nice ending. My question is about vaccination. I was just wondering, one of the challenges we had with COVID-19 was when we had to shift our messaging a bit that the vaccine protects from severe illness, but not necessarily from infection altogether. Do you anticipate it to be a similar situation with vaccines for H5N1? And is that something we should be a little clearer about going into this? Or and maybe this is speculative, but I just wondered if you had any thoughts about, how much we can actually stop infection with the vaccine for H5N1 versus it being more of a preventing severe disease.
Sure. Yeah, that's an excellent question. I think, based on what we know about the seasonal influenza vaccine, we do prevent deaths and hospitalization with that vaccine every year. And that's a really important central message. I certainly would expect that we would, at the limited information we would have, right, based on a hypothetical H5 virus evolving into people and really causing potentially severe disease. It really all depends on that clinical severity. I would expect that at the lowest level of expectation would be also to provide that protection against death and hospitalization. That said, there are, instances where we prevent burden, right? And so we track that every year for seasonal. It's hard to say what a vaccine for H5 might look like, but certainly I would expect it would be as good, hopefully, as the seasonal vaccine.
Dr. Pavia, University of Utah.
Yeah, thanks very much. And thanks for the presentation. And thanks for emphasizing all the things we don't know. As Nancy Cox used to say, you've seen one flu epidemic, you've seen one flu epidemic. So my question has to do with the translation of candidate virus vaccines into vaccine and the time it would take to do that, and what we would do in the case of a threat to the egg supply, which could easily happen, of course, with this virus. Do we have an idea of what the triggers would be to begin production and the impact that would have on seasonal vaccine? And given that it would probably take a long time to produce enough vaccine, I'm sure we have, but do we have a new and updated plan for prioritization in place? And that may be, something that other partners can chime in on as well.
Sure. Thanks so much. Yeah, I think we probably have the right speakers on this call and understanding the timelines involved. As I mentioned, that candidate vaccine virus is the precursor, right? It's your start material. And so a lot of steps have to happen in that space. And I'll defer to Dr. David Boucher from ASPR on the timelines there. Hey, David. But I can talk about the prioritization. I think that's a really important point, when we think about what would kind of push us into this space and thinking about a vaccine, making that available. We have a framework. We have interim recommendations for influenza in an influenza pandemic of how we would, again, kind of from a general perspective, tier individuals at the highest risk. That said, it's just a framework. It was utilized as a starting material, I think, for COVID-19. I think it's been used for other pathogens. Every bug is a little different. If H5 were to emerge and become widespread in people, we would look at the epidemiology, the laboratory data. We would really need that virology to help drive tailoring those priorities as far as who would be at highest risk for infection. But that's all public. We can certainly share that information. But of course, again, it's just a framework that we would think about. And what the changes that we would look for, I think off the top of my head, of course, we're looking for any changes that would suggest more mammalian transmission, change the risk to people that the virus is changing to be better adapted to human-to-human transmission. Again, clusters of cases. Epidemiology linked, not epi-linked, lots of different, there's no formula, but I think that we're all on the watch for anything that might change in the genetic space and in the epi space that would make us more concerned. And then, of course, talk to our flu risk management meeting experts to think through this and what we'd recommend. We rapidly convene them often.
Yeah, thank you.
Thank you. Dr. Linfield, Minnesota.
Thank you so much. Thank you so much, Dr. Dugan, for all your work on this and for sharing your insights. I wonder if you can talk a little bit more about the global collaboration that you have in assessing these viruses. As we know, there is this big outbreak in migraine waterfowl, and just wondering if you can talk a little more about.
Sure. Yeah, no, happy to do that. As I mentioned, we're one of 7 collaborating centers for the World Health Organization for influenza. And so part of that role is to not only for seasonal viruses, but for potential pandemic zoonotic viruses, really assess all of the data and share that information as it becomes available. And so we are often talking with our WHO Collaborating Center leadership, WHO Global Influenza Program, to understand what's happening in the space, what other countries, other groups are seeing, not just from a genetic perspective, from an antigenic and epidemiologic perspective. And so it's not just the 2 annual meetings that happen each year that the WHO convenes to recommend vaccine viruses for the seasonal virus vaccine, but it's also recommendations come out also for all the zoonotic subtypes, mostly the swine influenza viruses, so novel influenza A viruses, but H5, H7, H9, and candidate vaccine virus recommendations. So they're officially updated and discussed twice a year, but that is all still happening under the table behind the scenes. So where data are being shared and making sure that we are sharing specimens as well as data and information so that we can all be as prepared as we can be.
Great question. Dr. Dugan, thank you so very much, and extend that to your team as well. This concludes the 3rd session of our symposium. We will now take a 8-minute break. This will give you an opportunity to stretch your legs, have a snack, get some fresh air, return a phone call. Please don't go very far. We'll see you back here in 8 minutes. Welcome back, everyone. Thank you for rejoining us. We have 2 remaining sessions of this symposium, and I will remind folks this is being recorded. Panel or session 4 will examine the development of HPAI countermeasures. We are joined by 2 panelists for this particular session. The first is Dr. David Boucher. He's the Director of Infectious Disease Preparedness and Response at ASPR, the Administration for Strategic Preparedness and Response. Our second presenter is Dr. Michael Eisen, Chief of the Respiratory Diseases Branch at NIAID, the National Institute for Allergy and Infectious Diseases. I believe Dr. Boucher is going to go first. Let me hand it over to you, Dr. Boucher.
Sounds great. Thank you very much. Good morning, everybody. Before I get started, I just want to say thanks to ASTHO, IDSA, CST teams, and everyone else that brought us together. For all of you, though, for the interest you're showing by joining us here, I got to say, it's really heartening to know we're truly focused on developing the best response we can, and really glad to be here with everybody today. For the next slide, just want to touch quickly on some of the prior efforts that we can leverage. Specifically the procurement of personal protective equipment, PPE, and antivirals. Both of these are available in the Strategic National Stockpile, which is managed by ASPR. Regarding general availability, these products are obviously available on the commercial market, and this is the first avenue of sourcing. We are not tracking any supply chain issues for any of the items that I'll be discussing here. That said, want to make you aware of a few safety nets we have in place at the federal level. Before that, personal protective equipment is often available also at the state and jurisdictional levels, so please be aware of that. At federal, my colleagues at the U.S. Department of Agriculture manage the Food Safety Inspection Service, or FSIS, and the National Veterinary Stockpile, or the NVS. Both of those also maintain a supply of PPE. Here at ASPR, we do have a significant supply of PPE in the SNS. Talking about goggles, face shields, gloves, a lot of other reserves that can be tapped into if we need to step in and supplement other sourcing channels. So all of this really to say simply that appropriate use of PPE right now really should not be unnecessarily limited by supply issues. Shifting over to antivirals, I think you've heard earlier in the discussion from Dr. Dugan that the virus that we're tracking right now does not carry any of the genetic markers that we would commonly associate with resistance to the available antivirals for influenza. What this means is that the use of these antivirals for seasonal influenza would also be available for the treatment of infections caused by the H5N1 clade that we've identified in wild birds, poultry, and some mammals, including, of course, here, the focus, dairy cattle. We do have several of these antivirals in the SNS. We have oseltamivir. The branded name of that one is Tamiflu. We have zanamivir or Relenza, baloxavir or Zofluza, and then lastly, peramivir or Rapivab. Oseltamivir is the most commonly prescribed antiviral for influenza, and thus it is the majority of the SNS inventory. As with the PPE, we're not seeing any supply chain issues associated with these antivirals. And of course, we have identified just one human case of H5N1 associated with the spread among dairy cattle. We do, however, I just want to make everybody aware, we have tens of millions of courses of Tamiflu that can be used to supplement commercial sources and alleviate any supply bottlenecks if we need to do that. We did this during the 2022-2023 influenza season. That had an early peak that hit us a bit earlier than we're used to. It caused spot shortages of oseltamivir. We stepped in, made the federal supply available. If needed here, we would follow a very similar operational plan. So I'll switch gears to vaccines. Want to first focus on some really critical context for everything I'll go over here. We are very much in a preparedness posture right now. We're leaning into it, and we're advancing work that is actually quite normal for us under what we call the National Pre-Pandemic Influenza Vaccine Stockpile Program, or the NPIVS. Within ASPR, the Biomedical Advanced Research and Development Authority, or BARDA, leads the NPIVS and works in tight collaboration with CDC, NIH, FDA, and many other groups within the HHS and throughout the federal interagency. As we heard from Dr. Dugan earlier, our partners at CDC are constantly conducting surveillance to identify emerging influenza viruses. For those that are sufficiently new or concerning in any way, CDC builds what we call candidate vaccine viruses, the CVVs that Dr. Dugan mentioned, and then we can use these to manufacture candidate antigens that are tested for cross-reactivity to any viruses that we're interested in. BARDA then takes those CVVs and we work with our industry partners. These are the same pharmaceutical companies that manufacture the seasonal flu vaccine that I hope we're all taking every fall. We complete initial manufacturing of bulk antigen using these licensed platforms that our partners have. This lets us work out any manufacturing kinks. It also builds an initial supply of bulk antigen. In parallel with this effort, we also stockpile adjuvant under the NPIVS. This is that vaccine component that increases immunogenicity and allows us to get a good response with a smaller mass of antigen in each of the doses. These lines of effort would then converge in a response if needed and provide us with the building blocks of vaccine that allow us to jumpstart larger scale manufacturing. So also with the NPIVS, we sometimes do limited fills of the bulk and antigen, primarily for use in clinical studies of safety and immunogenicity. So that's really the what and the how of what we do all the time. The spotlight is obviously on H5N1 right now, but I want to just assure everybody that this work is constantly going on at BARDA and across HHS when the spotlight isn't shining. Dr. Pavia in the last section really asked the question of the day. What does all this give us now, and how does it translate into some of the things that you've heard about or maybe read about regarding vaccine availability? Spend a couple minutes just talking about that. As Dr. Dugan mentioned, we do have two CVVs that are well matched to the H5N1 virus that we're tracking in dairy cattle. Those are the two with the really long names. Commonly, we'll call those Astrakhan, American Widgeon. We have completed initial fill of some doses. We're working right now with our colleagues at FDA and our industry partners to determine if there's an appropriate regulatory pathway for use of these doses if we need in the current response. If there is, these doses would be the first wave of doses that would be used. At this level, we're talking about total quantities in the hundreds of thousands. This would most likely roll out in tranches of several hundreds of thousands at a time. I mentioned the bulk antigen manufacturing and adjuvant stockpiling. We have also completed that for these CVVs. If we needed additional vaccine above that hundreds of thousands that I mentioned, we could mix and fill this bulk material into final vials or syringes, and these doses would then be kind of that second wave of doses available. In this part, we're talking about up to about 10 million doses. If we needed to move beyond this, we would be able to leverage existing contracts with manufacturers and enter into large-scale production. This is where we would be able to build a supply of vaccines sufficient to provide doses to a wider swath of the country. As I mentioned at the top here, this is really a description of our preparedness posture. Everything that kind of put us into this crouched and ready position is supported by our annual appropriations and really part of ASPR's normal book of business. I do need to say that any pivot into response efforts beyond this would require additional resources beyond these. We are right now working with our colleagues at CDC, USDA, and other places within the federal interagency to ensure that if we need to pull any of these levers, we're ready to do so. Again, I really want to emphasize the preparedness aspect of all of this. Based on CDC's current assessment of risk to humans, vaccination is not a tool that is needed at this time. We do want to be ready if that changes, though. So that's really a very quick summary of the things that you might be hearing about in the news and in scientific conversations like these about the tools that we have here right now available to help out if they're needed. I want to pass over to my colleague, Dr. Michael Eisen. He can give you a look into the great work that our partners at the National Institutes of Health are doing towards that next group of influenza countermeasures. Dr. Eisen?
Great. Thank you so much. And again, thank you for inviting me to join you today. What I'll be talking about is investigational countermeasures. So those things that aren't available for us now, but hopefully will give us additional tools in the future. So really, I think that I'm going to just start with how the virus replicates, and this gives us a sense of where the various potential targets that we're considering. The influenza virus uses the hemagglutinin to bind to the sialic acid on the cell surface. This then allows the virus to be endocytosed. In the endosome, it goes through acidification mediated by the M2 ion channel, releasing the viral segments that are transported to the nucleus. There, the virus undergoes replication and onward production of viral proteins. This is where many of the various polymerase inhibitors would inhibit replication. The segments are then exported from the nucleus and packaged into virions that are bled from the cell surface. Before they can be released, they require the neuraminidase to cleave the connection between the hemagglutinin and the sialic acid receptors on the cell surface to release the virus. So with this, we can see the three key targets that are currently approved agents function through the M2 ion channel, the polymerase inhibitors, and the neuraminidase inhibitors. But many of the other potential components of viral replication are also potential targets for drug development. And through a number of grants, we are supporting efforts in the preclinical stage. And these are the efforts to get chemicals ready to be able to be applied as a druggable drug to enter early clinical trials. The areas that we are focusing on are hemagglutinin inhibitors, small molecule polymerase inhibitors, small molecule nucleoprotein inhibitors, and a number of small molecule endonuclease inhibitors. There's also a number of different efforts ongoing to identify optimized monoclonal antibodies that could provide passive protection and potential therapeutic uses. We also are investing in efforts to attack the host-acting antivirals, such as an SKI complex antagonist that would help inhibit viral replication. Likewise, there are two efforts that we're supporting to repurpose existing drugs. One is to create a stable lipid nanoparticle formulation of niclosamide, and another is to develop a novel delivery approach using micropatches for zanamivir. Additionally, we have one product being developed by Novo, which we are moving into early clinical trials with the first in human study of their product VTN-101, which is an orally bioavailable direct-acting small molecule active against the influenza nuclear protein. In addition, we are doing work to help support the use of medical countermeasures, including vaccines. One of the labs that we support through the SEER network, Richard Webby's lab at St. Jude's, had access to samples from a recently conducted study. In this study, in which they used 2 doses in a prime boost fashion of a novel H5N8 vaccine directed at the A gear falcon, the testing from the serum from patients that had completed 2 doses demonstrated that despite 16 amino acid differences between the HA from the current H5 and the gear falcon, the serology was able to identify cross-reactive antibodies to the A bovine H5N1 strain, suggesting that this may be a tool of moving forward. This is a vaccine that predates some of the ones that are moving forward, but again, I think highlights the work that we're doing to help support preparedness as well. With that, I'll turn it over to Joseph and the panel for questions.
Thank you so much, Dr. Eisen. Those were 2 excellent, succinct presentations. We have about 9 minutes for questions. I will ask our questioners, feel free to direct your question to either Dr. Boucher at ASPR or Dr. Eisen at NIAID. Let's go first to Dr. Pavia at University of Utah.
Thank you very much, Dr. Boucher and Dr. Eisen, for those great presentations, and for all the work you're doing. The work that you do when the spotlight isn't on is really important. And now that spotlight's on, it's all this is paying off. My question for you, Mike, is of the candidate technologies that you've invested in, which ones are closest to realization, both on the vaccine front, where we're looking at things like mRNA and self-amplifying mRNA and all the other platforms that NIH is invested in, and likewise on next generation antivirals.
Yeah.
Because hopefully we have a few years or longer before we have to deal with H5N1 human infection. But what do you anticipate coming online?
Well, of the things that we're currently supporting, the majority are still in the preclinical phase. So we don't have efficacy data yet to really put some insights as to which is going to be better. That being said, what excites me is that a lot of the agents that we're supporting target novel targets for the antiviral. And I think our goal in the long run is to have multiple agents that have activity against various targets, because while thankfully we don't see resistance to any of the available agents for this virus, if resistance were to emerge, we would want to have as many countermeasures available for us. In terms of vaccines, I think that there's a lot of enthusiasm for newer vaccine technologies. And in the seasonal front, there's a lot of effort moving forward. And hopefully, we'll be able to leverage some of those technologies for seasonal vaccine in the near future. We also support a number of programs through individual grants and through a large program, the Civics program, to really identify newer, improved vaccines over our current vaccines. And while I didn't present anything on that program today, we are supporting a wide range of vaccine approaches, including those that such as mRNA and other nucleic acid-based technologies that really would facilitate rapid updating and rapid application of vaccines.
Thanks.
All right. That's great information. Thank you. Dr. Cassandra, North Carolina.
Great. Well, thanks again, Dr. Boucher, Dr. Eisen. Again, just want to echo great information. Thanks for all the preparedness work that you've been doing all along. Really appreciate the information shared. And Dr. Boucher, I wanted to ask, you described very nicely the 3 phases of potential vaccine development. The first starting off with, tens of thousands of doses in those pre-filled syringes, all the way to full-blown production and ramp up, which would require additional resources at that point. Can you talk a little bit more about what the implications would be of those 3 stages for seasonal influenza production, and what priorities or what impact it would have for that as well?
That's a great point. So I mentioned we partner with the manufacturers of the seasonal influenza vaccines. The great advantage that gives us is that we're using licensed platforms. We can quickly pivot to a different clade and plug it into that. It makes the regulatory path, a lot of the manufacturing, a lot easier. The downside of that, though, is that it does leverage those same lines that are used for seasonal production. So the things that we're weighing right now when we're trying to evaluate the decision of when to go, it is obviously the situation on the ground. The risk to humans is first and foremost, the resourcing required to do it, and then the potential side effects if we disrupt seasonal production. We would obviously not want to do anything that causes shortages of the seasonal vaccine. So those are all the things that we're weighing right now. And it's you can see there's a lot of variables that go into making that decision. It's a great point.
Great question. Thank you, Dr. Boucher. Dr. Linfield, Minnesota.
Thank you. And my thanks as well to Dr. Boucher and Dr. Eisen and all the folks that work with you. I'm wondering if you can comment on what the triggers would be to do the safety and immunogenicity studies using the candidate vaccine viruses.
Yeah, so actually, fortunately, one of the other things that we do as part of the NPIVS is safety and immunogenicity studies. So we do have 2 studies that we're supporting right now. Those are being carried out by 2 of our industry partners. I don't know the clinical ID offhand, but they're both on clinicaltrials.gov. So we are doing both of those right now.
Wonderful. And one additional question, if I may, for Dr. Eisen. I'm wondering, can you comment on what is happening with the efforts to make a universal influenza vaccine?
Yeah. So again, one of the huge priorities of NIAID is to advance efforts to have a universal flu vaccine. And from our perspective at NIH, we define a universal flu vaccine as a vaccine that it marks a significant improvement over existing technologies. Those that protect longer, protect underprotected populations, and ideally would protect a broader swath of viruses so that you could have one shot and be protected against all potential viruses. We have a very large program that funds individual grants to investigators that are developing vaccines, and we are supporting a large number of candidates with almost every technology that can be thought of for vaccine production and implementation. We also have efforts in collaboration with our sister division DATE that's looking at some of the newer adjuvants to see how we can bolster and improve responses to existing and candidate vaccines. And as I mentioned, really our most important network is the Civics Network, which has 3 different discovery centers that are constantly working to discover new potential candidate vaccines, again, using a wide range of approaches, moving that into a manufacturing core and then on into clinical studies. And the goal of this is using the outcomes from the clinical trials to go back to the bench and really optimize the vaccines. So it's a huge effort, one that we're making progress on and are supporting through a number of different efforts.
Thank you, Dr. Eisen. Great question. If any other discussants have questions, please raise your hand. I see Dr. Hahn. Go ahead.
Yeah, thank you. I guess this is maybe a question for either of you. There's no point developing a vaccine that people don't want to take. And I'm just wondering, with some of the lessons learned from, challenges during COVID, kind of what are the plans to change our approach or to, it's early in the game, of course. We don't even know yet whether we will need to use one, but just thinking ahead, what are some of the different approaches that we will be taking to try to encourage the public to consider this vaccine if it's something that's recommended?
Dr. Hanna, that's a great question. Mike, if I'll take a first stab. Really you can cover anything I might miss. It's just such an important point. It's great to have the tools. They have to be used. We see a lot with the mis and disinformation. We're working closely with our partners here in HHS. We have ASPA, a lot of the groups. CDC does a lot of fantastic messaging about vaccines. I particularly liked in the last session, we were talking about effectiveness of the vaccine and how we're measuring that. Is it to prevent infection only? I don't think so. Preventing severe disease is obviously a great gain from vaccines. I think we need to lean into that a bit more and really be tighter on our messaging around all of this. But it's such a great point. Mike?
Yeah, and I think the other thing is the need for research to better understand how misinformation is spread, how that affects decision-making for vaccine uptake, and then more importantly, how different forms of media actually fuel some of that information. And there's programs that have been started by CDC and others that are really hoping to dig into this, because understanding the how and the why will help us best combat the real tide of pushback that we're seeing with vaccines.
I think we could have a whole nother symposium on many of these topics. We have time for one final question of this session. Turn back to Dr. Pavia.
So question for Dr. Boucher, but you'll probably want some input from all of the ASTHO and CST representatives on this. With antivirals, one of the big things we faced is the last mile problem, getting them from commercial and SNS stockpiles actually to the place they get dispensed within 48 hours. Can you tell us a little bit more about some of the progress we've made there?
Yeah, it's a great point. We face this not just within COVID, but also in the recent Mpox response, distribution of TPOXX, Jynneos, a little bit different there. But one of the things we're really trying to focus on is that, fortunately here, we are backfilling products that are already commercially available. I think we can do that in a way that doesn't put extra burden on our local and state colleagues to rely on that commercial distribution, be able to backfill with enough distribution points, enough destination points, so that we can get those and ready to backfill as best possible. Obviously, something that we look at as we scale a response. And again, I don't think we're going there, but in a mass distribution campaign, there are a lot of levers we can pull to tighten up and do some of the things that we did during COVID, test to treat, a lot of different tools, a lot of different lessons learned that we can employ.
Great, thank you.
Mm-hmm.
And I agree with you. I think it's highly unlikely that this will be a mass casualty event like an anthrax. This is more maintaining a supply through a long, difficult epidemic.
Exactly.
Great, excellent session. This will conclude this session. Dr. Boucher, Dr. Eisen, we thank you so very much. It's very helpful to hear what ASPR and NIAID are doing in the background, obviously, and you know, work that happens every day, whether we're dealing with an event or not. So thank you so very much. We will now go to our fifth and final session of this symposium. We will evaluate milk safety. And we're pleased to be joined by Dr. Donald Prater. He's the acting director of the Center for Food Safety and Applied Nutrition at the FDA. Dr. Prater?
Thank you very much for that, Dr. Kanter. And just as I get started, really echo those comments, appreciation to ASTHO for organizing the symposium and giving us some time to describe some of the key research questions and data gaps that we're looking at. Of course, the FDA is looking at the evaluation of milk safety. We have a milk safety system. So in the federal state milk safety system, nearly 99% of the commercial milk supply that's produced on dairy farms actually comes from farms that participate in our Grade A program and follow something called the Pasteurized Milk Ordinance. So the Pasteurized Milk Ordinance is a model ordinance that's embedded into many state laws and requirements, and it includes those controls that are necessary to help ensure the safety of dairy products. Importantly, it includes the pasteurization process, but it also includes other provisions such as the fact that milk from abnormal milk or milk from sick animals should not enter the milk supply. And this has been one of the key provisions that we've looked at as this situation has emerged. So as we've been evaluating this, of course, it's the first time that we've seen avian influenza in cattle. And so there are not a tremendous number of studies that have looked at pasteurization of bovine milk. There are some important key questions that we're asking. And so one of the questions that we're very interested in is what level of virus might be leaving the farms from apparently healthy cows. So you've heard some discussion already about symptomatic cows, sick cows. The milk from those cows should not be entering the milk supply. They should be pulled aside and that milk diverted or destroyed. Talk more about that going forward. Another question, well, let me come back to that one. So one of the challenges is taking a look at healthy cows and considering asymptomatic or convalescent animals and what level of viral shedding might be present there. Next question, does any live virus survive the pasteurization process? As I mentioned, there are not studies directly looking at avian influenza in bovine milk. We do have a number of other studies that are similar, looking at envelope viruses, but this is a key question for us. What level of shedding is in asymptomatic, pre-symptomatic convalescent cattle? So I mentioned that. And then, of course, the relevance there is what leaves the farm. That's the first question. Do different methods of pasteurization and dairy production have differing effects on the viability of H5N1? One of the things that I think we're acutely aware of is that there's a whole range of different dairy products. So we have everything from fluid milk to cheese to cottage cheese, sour cream. Most of these products are made with pasteurized milk, but it's important that we look at different components of dairy products. We also look at different methods of pasteurization. So that's another key scientific data gap that we want to fill. Critically, what is the risk of infectivity to humans via oral consumption? And so this is a key question that would underpin any sort of risk assessment is how much agent would it take to become an established infection? Therefore, what type of pasteurization criteria do we need to look at the acceptable public health outcomes? More studies that are needed. So what are some of the types of studies that we think are probably needed? Well, epidemiologic studies of transmission of the disease and the disease course in cattle, really important. So as these animals become infected, what does that profile look like for viral shedding? How can we identify asymptomatic, pre-symptomatic, and convalescent animals to minimize the amount of the agent that might be going into the milk supply? Other studies characterizing the level of live virus in raw milk leaving the farms. So we know that when milk leaves the farm, it is raw milk and could potentially contain some hazards which pasteurization is aimed to address. But being able to characterize this is really important. So we have some information about what kind of levels that we might expect to see in clinical or sick animals. But then as the milk from the healthy cows is aggregated, what does that look like as it's leaving the farm? Other studies talking about viral stability and persistence in milk. This is really important as well. Critically, and I think as some folks have already started to focus on, validating the pasteurization methods for H5N1 in raw milk samples in benchtop tests. Many of the tests that we start with to be able to inform this type of work are benchtop type tests. And so I'll speak a little bit more about those, but these are some of the important scientific studies that we believe are needed. Also validating pasteurization criteria in spike milk samples simulating real world levels leaving the farm. with a pilot pasteurizer. So we know that the benchtop studies contribute important information, but being able to look at the types of systems that are used in industry for pasteurization would provide additional information about the pasteurization criteria. So there are ways to look at pilot scale pasteurizers and to be able to look at them at the various time and temperature combinations. And I'll just mention here that there are a number of time and temperature combinations that are embedded in the Code of Federal Regulations and the Pasteurized Milk Ordinance. So all dairy products, all milk is not a monolith. There are a variety of dairy products. And similarly, there are different criteria that are used for pasteurization. Being able to assess whether any level of live virus is present in retail milk. And so this is important. We are looking at the success of pasteurization at the endpoint. I think we've been talking recently about the fact that PCR positive levels aren't necessarily indicative of live virus. And so it's important to be able to understand what we're seeing in retail milk. As well, research on routes of infectivity of H5N1 in mammals. These are important experiments. I'll move quickly on this because I know we have a number of experts that understand the methods that are being used. But importantly, it does not mean that the sample contains intact infectious pathogens. And as a result, we need to do additional testing to determine whether intact pathogen is still present and remains infectious. In order to do that, we're aware that there is a gold standard type of study. Embryonated egg viability studies are the ones that we look most carefully at as a sensitive detection for active and infective virus. This is one of the additional tests performed following PCR testing. And so it provides some information by injecting embryonated chicken eggs with a sample and then evaluating to see if any active virus replicates. And then an additional type of tissue culture that we're looking at are the Maiden-Darby canine kidney cell cultures. So what are the types of studies that FDA is participating in? There's a whole variety of studies that might be going on, some of which FDA are participating in. These benchtop pasteurization studies using spike milk and milk from affected cows. Also the pasteurization validation studies that I mentioned with a pilot scale pasteurizer. Determining viral load in raw milk that could be presented to processors. This might be different than what we see in a clinically infected animal. We would expect it to be. And then also a survey of avian influenza and retail products, as I mentioned before, followed by viral isolation in eggs. These are some of the studies that FDA is looking at in partnership with our colleagues at USDA Agricultural Research Service. So the pasteurization validation studies, we are looking at some of the most common time and temperature pasteurization combinations, including high temperature, short time. This is a method that uses 72 degrees centigrade for 15 seconds. We're also looking at this in both spike milk samples as well as samples from clinically infected cows. And then the other type of study that we're looking at, I mentioned before, involves a pilot scale continuous flow pasteurizer, and it will be informed by some additional data that we're doing in a double-blinded study. So why do we need to determine that? Because there will likely be a different viral load that will be encountered by the pasteurizer. And so we are working on a double-blinded study utilizing some samples that are collected under our grade A program. They're sent to a third-party lab, and then ultimately for blinding, and then ultimately they will go to the USDA ARS lab where we will follow that same procedure, looking at PCR detection followed by quantification in live eggs that will help us to generate the information that will inform our pasteurizer study. Retail dairy product sampling. So we are looking at retail samples of various milk and milk products. And again, hopefully the USDA ARS lab is conducting the quantitative PCR, looking for detection of genetic material, and followed up by quantifying for any potential live virus via an egg culture. Also on this landscape, so I mentioned that FDA is working on this. Our colleagues at NIH through their investigators are working on this. There are important questions to be answered, looking at viral levels in different fractions of milk, so the skim milk versus the cream, looking at potential ingestion of raw milk through from affected cows and rodents, some work on pasteurization validation, very similar to the benchtop test. There's other work ongoing at universities, and then importantly, as have already been discussed, APHIS epidemiologic investigations and the CDC state epidemiologic investigations of farm personnel will also provide important information.
Great. Dr. Prater, thank you so very much. It was an excellent presentation. I particularly appreciate you highlighting clearly the areas where we need more knowledge and that there's work ongoing. So thank you so very much. We've got about 9 minutes for questions. Let me go first to Dr. Harris in Alabama.
Good afternoon, Dr. Prater. Thank you so much. That was terrific, and we just appreciate the work you and your team are doing. You've been great partners with us in public health as well as with everyone else through pretty difficult times, I'm sure. So thank you for that. I wanted to ask you a question about the testing that's been done to this point. You know, we saw I guess, really reports a couple of days ago about genetic material found in samples, and I think the language was samples with an S in different parts of the production chain. And I don't know to what extent you're able to talk about that, but it helps our communication, you know, and how we do our messaging to the public about food safety. And I wonder, do you have any more details? Was this directed testing from producers that were somehow linked to where these outbreaks were occurring with cattle, or was this truly random testing, or just, you know, sort of spot checks of products, or what are you able to tell us about that?
Yeah, thanks, Dr. Harris. We're just looking at, I think you're referring to the retail milk sampling testing. And so we've just received some results. This is a scientific symposium, so y'all will be familiar with the fact you get late breaking results. So we've just received some results a few minutes ago, and we're still analyzing them. But at a top level, it looks like about 1 in 5 samples, maybe with some preponderance for areas from known herds. And so we'll have more to say about this as we complete our investigation. This is just coming in this morning, in fact. And I just want to remind that the PCR positive results representing viral fragments would necessarily need to be analyzed to determine whether there represents any viable virus that pose a risk to consumers. It's important for us to get this information. Also, we've reminded that early work by the NIH investigators indicate an absence of viable virus in their studies of retail milk, so that's helpful. And then, of course, we're following very closely CDC's food safety group as well as its surveillance team to make sure that they're monitoring any emergency department data, flu testing data for any unusual trends. And thus far, we haven't seen anything come up there. So overall, even with considering these results, we haven't seen anything that would change our assessment that the commercial milk supply is safe.
Thank you.
Thank you. That's great information. Dr. Moore, North Carolina.
Yeah, thank you so much, Dr. Prater. It's really great to hear about some of the milk sampling that's already underway or being planned. So I know we're all looking forward to hearing more about that, particularly from the herds where there aren't any symptoms. I guess you did mention some really interesting things about NIH-funded work looking at the risk of in rodents. I think you said, we're all used to thinking about the risks of raw milk being primarily gastrointestinal pathogens. And so I wonder if you have anything that you could share about current knowledge about the risk of influenza virus infection from raw milk consumption, or clearly that's being worked on, but if there's anything that you could share about what's known now.
Yeah, thanks, Dr. Moore. That's sort of two good questions in one. We do have advisories already in place for raw milk. We are aware that raw milk does contain a variety of hazards, pathogens, and we've advised folks that people do not consume raw milk. It's not allowed in interstate commerce. I think that our advice stays the same in the case of this particular pathogen. You mentioned about route of ingestion and that versus an oral route. That is one of those scientific gaps we would like to have some more information on, because that will definitely inform our risk assessment. We know colleagues under the NIH research might be looking into that, but that is going to be an important piece of information as we do our risk assessment here.
Thank you. Great question. I should also note we do have Dr. Hillary Marston, the Chief Medical Officer for the FDA, on the line as well, should we need her expertise as well. Let's go to Dr. Rexak, Mount San Antonio College.
Thank you very much for an excellent presentation, Dr. Prater. I have similar questions regarding the transmission of HPAI via milk. As in the first session, we heard from the veterinarian who spoke that it does get transmitted between cattle by milk. And so my question would be, how long does the virus remain viable in milk? And also, you mentioned infectious dose. Do you have any idea about what that infectious dose might be via milk?
Thanks, Dr. Rex. As I highlighted in my presentation, those are two really important data gaps that we have. And so I think the folks that are working on the animal-to-animal transmission are looking at how that's occurring. So I would defer to the APHIS veterinarians on that. But with respect to infectivity, I think that's another place that we're looking. So there is related information, but I think that's still a data gap. And I know that colleagues at NIH are focused on that as well. So it's a good question. It is a data gap at this point.
Great question. Thank you. We do have a few more minutes left for questions. If you have a question, if you are a discussant, either currently spotlighted or otherwise, please go ahead and raise your hand. Dr. Harris.
Yeah, do you have any recommendation at the moment for milk that's been condemned and removed from the supplier about how it can be used or how it's disposed of or anything like that?
Yeah, thanks, Dr. Harris. At the present time, our recommendation is that any milk that is discarded be heat treated. And so whether it's being discarded in some way or fed to other animals, that this needs to be heat treated at temperatures that resemble commercial pasteurization. And again, I would just say really important that be done, because we know that such milk could serve as a reservoir where animals could become contaminated. So being able to treat that milk so that that doesn't occur is really important. That's our recommendation, that it be heat treated.
Dr. Rexak.
I'm really unfamiliar with the way in which the testing and everything is done, but I'm assuming that a lot of it is done on large facilities, large dairy facilities. Is there anything being looked at in terms of the smaller facilities where you often have mixed populations, like you might have chickens and you might have dairy cattle? Because I know when I was working in Central California area, there were a lot of small farms where they would typically sell raw milk. And they had mixed herds and things, and what the risk of transmission might be in a setting like that.
Yeah, it's a good question. I know that the regulations on raw milk, that it is allowed within a state, and so those regulations are governed by the state that allows that to occur. So I think, hopefully they are looking at that as well.
Great, thanks. Dr. Hahn, Idaho.
Yeah, thank you so much. I have a question about cheeses. My understanding is that even dairies that are, pasteurize their milk, all their milk for production, may be and I'm not an expert in this area, but some of that milk might be used for cheeses without pasteurization, because the process of making cheese is thought to provide safety to the public. But could you address that issue, because it is not a pasteurized product, to my understanding, in some cases?
Yeah, thanks, Dr. Hahn. Raw milk cheese, we are aware that some states, again, allow that. We do allow raw milk cheese with 60-day aging in interstate commerce. And so we're looking at that. By the time you get out to 60 days, there are a number of factors that need to be considered. And so we are taking a look at that as well. But it's a good point to raise that raw milk is not just consumed as milk, but it is also potentially consumed as raw milk cheese. Thank you.
Thank you.
Great, thanks. That's a great question. We will conclude this and final session there, Dr. Prater. Thank you and your colleagues at the FDA so very much. We appreciate the work you do always. This concludes the fifth and final session of our symposium. Let me take 1 minute just to express my deep appreciation for the federal partners that joined us today. We had partners from ASPR, CDC, USDA, FDA, and NIAID. We deeply appreciate the work that you do and are grateful for the insights you shared with us. I'll tell you in all honesty, I learned so much during today's symposium. It was sometimes challenging for me to keep up with my duties as moderator. This was a ton of information. I really do thank you for what you shared with us. I also want to express my appreciation to our partners in hosting today's event, the Council on State and Territorial Epidemiologists and the Infectious Disease Society of America. Thank you for the work you do, and thank you for your partnership. If there are media inquiries that are specific to one of the topics that were presented on today, I would encourage you to direct those media requests to that respective agency. If there are any other media requests on anything else about today's event or otherwise, feel free to send those to us at ASTHO, and you can email them to media@astho.org. That's media@astho.org. A message to public health practitioners on the ground. Thank you from the bottom of my heart for what you do. The work you do matters on this issue and many, many others. So thank you. I hope you all learned a decent amount today. I hope this was worthwhile, and I hope you have a really good rest of your day. This is ASTHO signing off. Goodbye.